So let's talk about C15. As shared, this is the first essential fatty acid to be discovered in 90 plus years. When we initially made that discovery of association in dolphins, we then moved pure free fatty acid C15 into the lab. And over 3 years, we did 8 studies, guided in part by Dr. Ed Dennis, who's a leader in fatty acids. He was the editor-in-chief of the Journal of Lipid Research for 15 years. And he said, gosh, here are the series of studies that we should look at to see if it's a beneficial and active fatty acid. And we found not only is C15 an active and beneficial saturated fat, but it was meeting these rare criteria of being essential, which means our bodies require adequate levels of C15 to maintain our physiological health, but our bodies can't make enough of it. That means that we have to get certain amounts of C15 from our diet. The dolphins needed adequate amounts of C15 from their fish. As we'll learn with people, we need to get adequate amount of C15 from our diet, and our primary source is whole dairy fat. So you can see C15 is such a simple molecule. It is 15 carbons that are all together, no double bonds, so it doesn't bend. So it's literally this tiny little 15 carbon chain, super simple. Nature has made this molecule essential nutrient to do a lot of really important things that we'll talk about. So it is a dietary odd chain saturated fatty acid. As I mentioned, it's a stable fatty acid with no double bonds. That means it's resistant to lipid peroxidation. And we'll talk about one of its most important points is it strengthens cell membranes. So we published this in Scientific Reports in 2020 and made the case that this was an essential fatty acid. So what the paper we just came out with is we've now taken the past 10 years of research that we've been doing on C15 and brought it all together along with the wealth of data from other folks around the world showing that C15 reverses ferroptosis and effectively stops all of the core components. We and others have been able to show that C15 gets into our cell membranes, it stabilizes and strengthens our cells by 80%, specifically in red blood cells. It prevents them from hemolyzing. It lowers lipid peroxidation. It lowers intracellular iron. We'll talk about that mechanism of action later. It has been shown to lower reactive oxygen species by 45%. And as a result, it repairs mitochondria and it restores healthy cells. So if you have a molecule that goes in and truly helps with each part of this process, this is now starting to point at this is not just a fix, that having too low levels of it is likely driving the cause of ferroptosis. So in addition to ferroptosis, which then we again propose in our paper that this is the cause of ferroptosis, a primary is C15 deficiencies. C15 has multiple mechanisms to enhance health and longevity, so it is a pleiotropic molecule with these activities with broad effects. So this includes anti-inflammatory, antioxidant, antifibrotic, anti-cancer, and antimicrobial. We'll share in a little bit this suite of benefits all shown in a dose-dependent, significant fashion in the peer-reviewed literature. Matches that of rapamycin. So we'll get to that in a little bit. So when we look at its specific receptors and mechanisms of action, it has these wonderful suite of longevity enhancing activities to the right by activating AMPK, inhibiting mTOR, which are key in the longevity pathway. mTOR obviously is the target of rapamycin. AMPK activation is the mechanism of metformin. In addition, we've been able to show that C15 activates PPAR alpha delta. These are known receptors, well known to help regulate metabolism and immunity, right? PPAR alpha activators are how fibrates work to be able to decrease triglycerides and even LDL cholesterol. It's also an AKT activator, another win for glucose control, and it inhibits both HDAC6 and JAK-STAT. JAK-STAT is my favorite mechanism with regard to anti-inflammatory, broad anti-inflammatory mechanisms, and both HDAC6 and JAK-STAT have been demonstrated by others are the mechanisms of action that explain C15's anti-cancer properties. We have not been doing much in that area. A little bit, it's just whole other teams are working on that component and potential for C15. So when we move to beyond mechanisms of action, we then have how C15 has clinical efficacies that improve health. And this includes double-blinded and controlled clinical trials. When we look at the animal studies, C15 treatment, oral C15 provided over 12 weeks, effectively alleviates anemia, lowers liver enzymes, lowers cholesterol, glucose, alleviates liver iron deposition, and decreases liver fibrosis in animal models of type 2 diabetes, as well as fatty liver disease and NAFLD. Importantly, this holds true for a key model for ferroptosis, that is a rabbit model that C15 had remarkable abilities to reverse, just like we had seen in the dolphins. When we move to people, beyond the epidemiological studies, we now have 2 randomized controlled double-blinded clinical trials with pure free fatty acid C15 given orally over 12 weeks. One study was done in Singapore, and it was with women with a history of NAFLD, and the other is a study that is soon to be published. So excited for that to get out of young adults who were overweight and had a history of NAFLD as well. And what these studies showed was that a C15, oral C15 effectively raised hemoglobin. And again, we'll talk about these parallels, right? Of alleviating anemia, strengthening red blood cells, alleviating anemia in animals, raising hemoglobin, strengthening red blood cells in humans. Importantly, lowers liver enzymes, lowers LDL cholesterol, and improves the gut microbiome, which includes growth of a gut microbe called Bifidobacterium adolescentis, which is super fun because it's basically our core microbe that helps with regard to metabolic health. But it's also been shown to extend longevity in worms and flies. So might have a more interesting twist that it helps with the growth of these microbes. So have I mentioned? So C15 is the first essential fatty acid to be discovered in 90 years. Once we discovered that C15 was an essential fatty acid, we then wanted to test it head-to-head against pure omega-3, the highest performing, specifically EPA, and when we're talking about heart health. So we used this test this panel called BioMap, which basically looks at 146 different biomarkers across 12 human cell systems mimicking various disease states. So this allows you to test different pharmaceutical companies use this test routinely to assess not just safety, but efficacy for potential diseases. And so what we showed was that pure C15 had all of the benefits of pure EPA and then 3 times more. And additionally, it was safe. So because it has no double bonds, when it got to the higher doses of like 50 micromolars, it continued to have efficacy and be safe. Omega-3s, on the other hand, once it got to that 50 micromolar state, because of the lipid peroxidation, it actually started to kill cells. So an important part of attribute with regard to C15. We did the same test, so then we repeated it. As we started seeing that C15 was matching the benefits of rapamycin, we then worked with Dr. Nick Shork, who's the leader of the NIH's Longevity Consortium. And Nick said, this is looking like a geroprotector to me, and a great longevity enhancing molecule, especially with all the other molecules he is seeing going through, the anti-aging pipeline. So we did the same test with Biomap, and we went head to head against a carbose, metformin, and rapamycin. And again, C15 had these 36 benefits, which was better, far better than acarbose and metformin, and was mighty close to rapamycin. So again, not a surprise that C15 and rapamycin had similarities based upon the fact that both of them have all of these activities on the right. So our conclusion from that paper, which we published in Nutrients late last year with Nick, was that rapamycin might actually be just a C15 mimic, right? And we didn't actually have to go to Easter Island to get this molecule that's made by a foreign bacteria, and is very complicated and comes with a really long list of side effects. Instead, what we've really needed for longevity is C15, something that every mammal gets from birth in mother's milk. So it just has taken us a while to get there, and thanks to the dolphins to helping us discover it. So when we talk about we're moving into the longevity realm, that C15 targets 6 hallmarks of aging. The number keeps increasing the more the world has been working with it. Which shows that it slows aging at the cellular level. It slows cellular senescence by inhibiting mTOR and strengthening cells by 80%. We talked about it repairs mitochondria and lowers reactive oxygen species by 45%. It stops inflammaging by lowering these 18 pro-inflammatory cytokines. It restores cellular signaling, all those important mechanisms of action that I talked about earlier. The new ones that came in were the gut microbiome, as I had just shared, and a really fun study showing that there's an association of C15 with slower biological aging, a study that showed that people with higher C15 levels had lower DNA methylation. And then when they applied it to these epigenetic clocks, they showed that the higher the C15 people had, the more likely their biological age was younger than their chronological age. So association, but we have a lot of reasons to believe why that C15 may actually be causing it versus just association. So the next piece with regard to longevity is talking about the cell membrane pacemaker theory of aging. And this was introduced by A.J. Holbert in 2005. And he came out with this theory where he showed the greater the cell membrane stability, the fatty acids in the cell membrane, the longer a mammalian species lived. So he was able to show by looking at about 40 or 50 different mammalian species that the more saturated the fats in the cell membrane, the longer a mammalian species lived. So we have much higher levels of sturdy saturated fats compared to mice, and then everything in between. So really exciting theory that matches the mechanism, a core mechanism of C15 with regard to ferroptosis and cellular stability. So what we introduced in this paper in Metabolites from a couple weeks ago is the cellular stability hypothesis, where we're taking that initial hypothesis theory and we're pushing it to say the greater the cell membrane stability, the longer an individual's longevity. So we believe that because of the evidence to date that we can push C15 levels, we need to get them at adequate levels, and that can help dictate the strength of our cell membranes and to help support, to stave off chronic diseases to extend our longevity.